Hi everyone. Um, my name is Jillian, and this is my tutorial project titled Understanding the Relationship Between Promoter Methylation and TP53 Gene Expression using the UCSC Genome Browser. So, the goal of this project is to explore how promoter methylation might regulate the expression of the TP53 gene, which encodes for the P53 tumor suppressor protein, which is a key player in cell cycle control in apoptosis. And so I utilize the UCSC Genome Browser to visualize data sets from the ENCODE project that show DNA methylation levels and RNA expression in two different human cell lines. GM12878, which is a lymphoblastoid line, and HeLa S3, which is a cervical carcinoma cell line. In this tutorial, I'll give a brief step-by-step on how I used UCSC to find the TP53 region, add methylation and RNA-seq tracks, as well as interpret what these visualizations can tell us about gene regulation. So, in this project, I used whole genome bisulfite sequencing to assess DNA methylation and then RNA-seq data to assess um, the expression levels of TP53, both of which are part of the ENCODE project, which I will demonstrate how to incorporate those filters in UCSC. So you want to first start by navigating your way to the genome browser. So it's genome.ucsc.edu. This is the main page it'll bring you to. We want to click on Genome Browser here. Make sure you have human selected, and this is the assembly you want, the GRCH38, otherwise known as HG38. We're going to click Go, and this is what you'll first see. It does offer a short little guided tutorial. I would definitely recommend viewing that if this is your first time seeing UCSC or interacting with it. It is very useful. So in this search bar at the top, right here, I'll type in TP53. I'm going to click on this one here. And you can see this is where it is. So at first glance, you can see it's located on chromosome 17. These are the exact coordinates in which it is located. You can tell that it's on the minus strand due to these little arrows going from right to left. So the promoter region is going to be located down here. So I'm going to slide this way. All right, so here it is. So the transcription start site is going to be around here. This is important because that's where we'll observe certain methylation patterns and expression on this gene. So when you scroll down, there's a bunch of different tracks with specific filters in each one. So under regulation, for example, down here, I'm going to select CPG islands to show and refresh. And you'll note that we do see a couple here that are located upstream and downstream of the transcription start site of TP53. Then under gene and gene predictions, I'm going to um, I'm gonna set GenCode and NCBI RefSeq both to full. Actually, I'm gonna set them to dense, so they're not super overstimulating. Um, and what that does is it just gives you a reference of other analyses performed with this of different variants across this gene, just for comparison. Um, so these blue boxes here, like these really thick blue boxes, those demonstrate the presence of an exon, whereas the presence of these skinnier lines demonstrate the presence of an intron. And so you can also um, add DNA methylation data from ENCODE. So I'm going to show you how to do that now. You're going to go to My Data, go to Track Hubs. To observe the DNA methylation, we're going to look at the ENCODE DNA track hub. So we're going to connect to that. And you want to specify which one we're doing, which is this HG38. Alrighty. So then you'll see that it added this hub down here as a custom track. And so the method I chose to focus on was whole genome shotgun bisulfite sequencing to observe methylation patterns. So I'm going to click on that. And then we only want to focus on CPG islands for now. So I'm going to click on both the replicates for the two cell lines that I'm focused on. Make sure down here they are set to full so that way we can see them displayed across our model and submit. And so now we can see them all here. You'll notice that when I move my mouse across these, a certain value comes up. Uh, and so that is the percent methylation. So for example, when I hover over a larger peak, it'll say something like this, 0.538. That indicates that in that specific position on the chromosome, about 53% of the reads are methylated there. And so you'll notice that certain regions, like right here and right here, are where there's a bit of a more densely populated area for higher peaks correlates to the location on that chromosome where CPG islands are occurring. And so next I'll add the RNA-seq expression data from ENCODE as well. So to do that, we're going to go to Bind Data again, and then go to Track Hubs. And then I'm going to go to ENCODE RNA Track Hub and connect to that. Again, select the right one. Alrighty. And so then we should see that one pop up right here. I'm going to just collapse this one so we don't get confused. And then we're going to go to RNA-seq by biosample so we can select the specific ones we want. And then they are in alphabetical order, so I'm going to find the GM128 here. I'm going to just click on it because there are several replicates for each strand. So we're just going to take the first replicate for now. If you're doing a more in-depth analysis, of course, it would make sense to observe all of the available replicates. But for now, we'll just look at those ones. So the specific encode ID was this one here because we're doing the poly A plus RNA-seq. And so we want to take the forward and reverse strand. So what I'm going to do is I'm going to just go through these and deselect all of them except for those. Make sure this is set to full and submit. And what we should see now are the peaks for that. Again, just again at quick glance, you can see the higher peaks typically represent higher expression or higher signaling. And so I'm going to go ahead and do that same thing real quick for the HeLa S3 line. And let's see. All right, again, I'm going to just select the first replicate for the minus and plus strand. The encode ID from this one was the CPR. Again, we want the forward and reverse strand. So I'm going to go through and remove all of them besides the one with those IDs. And we're going to submit that. And then you should see those ones pop up. Oops. Sorry, I don't think I set that one to full. Nope, I didn't. Let's select that there. All right, now we can go ahead and submit that, and we should see it show up. There we go. All right. So, again, like I mentioned, these tracks show read coverage, so taller peaks will correspond generally to higher expression. And so this pattern might suggest that higher promoter methylation in HeLa S3 uh, might suppress TP53 expression due to these higher RNA signaling expressions. And this can align with how methylation can silence tumor suppressor genes. So I'm going to zoom in a little bit more on the transcription start site of TP53. So I'm gonna just put in coordinates here. So I'm going to copy and paste this over on this side, but I'm going to alter the coordinates so that way we are focusing in more on the transcription start site. So yeah, you can see we're kind of in this range here, which is right where that is. I'm gonna click search and it should zoom us in a little. You'll notice that these will end up looking like this. Since we did just zoom in a lot, it'll look a lot more stretched out. And let's see. I'm gonna zoom out a little bit. All right. So here we are. I'm gonna just move it over. You can click and drag so you can get a little bit of a better representation. I'm gonna, oops, I meant to zoom in there. Wait for it to load. And then I am gonna zoom in by 10. And you'll notice that the thick boxes get a little bit thicker each time. And then you'll notice again, here is that first exon on the TP53 gene. Again, you'll notice, especially on the minus strands uh, of both of these cell lines, the poly A RNA signaling is relatively strong. And then when we go up here as well, you'll notice that where there's a bit of a larger clumping of these methylation states, they're all relatively located within this transcription start site region. In addition to this, you can observe things like DNase hypersensitivity to observe open chromatin, as well as looking at certain histone modification tracks to identify any potential active promoters. And so to get quantitative data from this, you can go up to Tools and then go to Table Browser. And then, so this is for if you want to perform further quantitative analyses. So I'll show you how to just download some of this data onto your computer. So you want to make sure you have all the appropriate things selected. You do have to go through and do this separately if you're just doing a couple individual tracks. So I'll just show this one for example. Once you click that first one, it'll update everything else so you can go through and select it. Since we did whole genome shotgun bisulfite sequencing, I'm going to select it down here. And then for the table, I'll just choose the GM128781, making sure I'm selecting a CPG island. Again, so there's two here because it's showing the forward and reverse. I'm just going to select this first one just to show you for an example. Make sure you have that specified position there because if you look at the whole genome, it's going to be a lot of data that will make it really hard to hone in on the region of interest. So it's important to specify the position. And then down here, you can send it to other databases like Galaxy and Great. But for today, I'm gonna just highlight the um, ways in which you just download it on your computer. So I'm going to download it as a browser extensible data file. So it's just plain text. And then I'll click Get Output. Yes, genome. Okay. And then Get Bed. Okay. So what you see here, this bed file includes the columns for chromosome, start and end position, methylation signaling, and then what strand it's located on. So the higher signaling values down this middle one corresponds to a higher methylation percentage. And so this process can be repeated with the other cell line and then for RNA-seq analysis. And in summary, um, using the UCSC genome browser, I was able to integrate and visualize methylation, expression, and regulatory data all in one platform. And so I was able to observe TP53 promoter hypermethylation in these cell lines, and this might suggest some sort of epigenetic silencing due to the increased methylation states near the transcription start site. While this does provide clear visual evidence, some limitations do exist. For example, certain promoter regions do have limited whole genome shotgun bisulfite sequencing coverage, and not all regulatory data are available for every cell line. So that might make it a little difficult to put things into perspective. But despite these challenges, UCSC does prove to be a valuable and accessible tool for exploring epigenetic regulation of genes, especially those with roles in cancer biology, like tumor suppressor genes like TP53. And with that, I would like to thank you for listening to my video tutorial, and I hope this helped you.